Genetic engineering is the process of manipulating an organism's DNA by adding, removing, or altering a gene to achieve a desired trait. It's like taking the genes responsible for the purple pigment in snapdragon plant and transferring it into tomatoes in order to have a purple tomatoes instead of the usual red ones. Once a new gene is added into the DNA, it's now called a recombinant DNA, and the organism itself is known as a transgenic organism or genetically modified organism. Gene transfer is not confined to a specific organisms. It can actually occur between different organisms from different kingdoms, like between plants and bacteria, or even bacteria and humans. The process of transferring genes occurs in a sequential manner. It starts off by identifying the gene of interest, isolating it, and make many copies of it till it finally reach the final host organism. In this video, we're going to talk about each of these steps till we reach the final host organism. Let's say we want to isolate the gene responsible for the purple pigment in snapdragon plant. First, we need to cut that gene out from the plant's DNA. To do this, scientists use tiny scissors called restriction enzymes. These enzymes were first discovered in bacteria, where they serve as a defense mechanism against invading viruses called bacteriophages. They recognize specific sequences in viral DNA and cut them at precise sites, restricting the infection. Bacteria that have these enzymes survive the attack, while those that don't eventually burst open and die due to viral replication. Because these enzymes restrict viral infection and cut within the DNA, they are called restriction endonucleases. Each type of restriction enzyme recognizes a unique DNA sequence and cut it at a specific point. For example, the enzyme PAMH1. This restriction enzyme can only identify the following set of nucleotides and their complementary sequence, as they read the same from both directions. Such sequences are called palindromic, and the enzymes are called palindromic restriction enzymes. Restriction enzymes can cut the DNA into two different ways, either a straight cut to produce blunt ends or a staggered cut to produce sticky ends. Sticky ends are especially important in genetic engineering because they leave free nucleotides exposed and ready to bear with another DNA fragment cut by the same restriction enzyme. When we cut a long DNA molecule, we end up with a mixture of fragments of different length. To find our gene of interest, we must separate these fragments using a technique called gel electrophoresis. Gel electrophoresis separates DNA fragments based on their size and charge by applying an electric current through a gel matrix. The gel is made of agarose, which is a polysaccharide derived from red algae. When heated and cooled, agarose forms a porous, jelly-like matrix that allows molecules to move through it, and then small wells are made in the gel, which is then placed in a tank filled with a buffer solution to conduct electricity. DNA samples are carefully loaded into the wells, and when an electric current is applied, the DNA, being negatively charged due to its phosphate backbone, moves toward the positive electrode. Smaller DNA fragments travel faster and further through the gel, and to visualize the separated fragments, the gel is stained with a DNA binding dye such as ethidium bromide, which fluoresces under UV light. But how do we know the size of our fragments? We use a DNA ladder, which contains fragments of known length, and by comparing our samples to the ladder, we can identify the fragment containing the target gene. And once identified, the desired DNA band is carefully cut out and purified from the gel. The purified DNA fragment is used to make multiple copies using PCR, short for the polymerase chain reaction. The PCR is a laboratory technique that rapidly amplifies a specific DNA segment, producing millions to billions of copies in no time. For PCR, what we need is the DNA sample, nucleotides, the tag polymerase, primers, buffer solution, and a Pendorf tube. The Tag polymerase is a heat-stable enzyme because it was originally isolated from the bacterium Thermus aquaticus, which lives in hot springs. It's the lab's alternative to the human DNA polymerase. While primers are short single-stranded DNA that guide the Tag polymerase where to start building. All the components are added together in certain ratios and then inserted into the PCR. PCR cycle occurs in 3 steps: denaturation, annealing, and extension. First, the double-stranded DNA is heated to 95 degrees Celsius to be separated into 2 single strands. The temperature then drops to 65 degrees to allow the primers to bind to their complementary sequences on the DNA. And finally, the temperature is raised to the optimal at 72 degrees Celsius for the Tag polymerase to start building the new DNA strands using the nucleotides and guided by the primers. This cycle repeats many times, exponentially amplifying the DNA. Now that we have enough of the desired DNA, we need a way to deliver it into the host organism. In order to transfer the desired gene, we use something called vector. Vectors can carry foreign genes into the cells. There are 3 main types of vectors, either plasmids, viruses, or liposomes. Plasmids are small circular DNA molecules found in bacteria that can carry foreign genes and replicate independently, while viruses are natural carriers that deliver genes into the host cells. Liposomes are artificial lipid vesicles that encapsulate DNA or RNA for delivery. In our example, we use the plasmid vector to transfer the Snapdragon genes into tomatoes. The DEL and ROS1 are the genes responsible for the anthocyanin pigments, which give the Snapdragon their purple color. These genes are isolated and cut using restriction enzymes that produce stickins. In the meanwhile, a TI plasmid is isolated from the Agaribacterium tumefaciens, a bacteria that naturally infects plants. The TI plasmid is cut with the same restriction enzyme to create matching stickins. The Snapdragon genes and the plasmid are joined together by an enzyme called DNA ligase, forming what's called a recombinant plasmid DNA. To get this recombinant plasmid back inside the bacterial cells, bacteria are treated with a calcium ion solution and then heat-shocked. The calcium ions make the membrane more permeable, and the brief heat helps the plasmid enter the cell. However, only a small fraction of bacteria takes up the plasmid, so we need a way to identify them. The plasmid should include a marker gene, either an antibiotic-resistant gene or a green fluorescent protein. If an antibiotic-resistant gene is used, the bacteria are grown on a medium containing that antibiotic, and only the transformed bacteria survive. If a green fluorescent protein is used, the transformed bacteria glow under the UV light. These successful bacteria are called transformants, and they are then used to infect tomato plants, allowing their recombinant plasmid to integrate naturally into the plant's genome. As the plants grow, they express the Snapdragon genes and produce purple-colored tomatoes instead of the red ones. To quickly recap, genetic engineering begins with identifying the specific gene responsible for a desired trait. Once identified, that gene is isolated and copied using molecular techniques. The copies are then inserted into a suitable vector, which helps deliver the gene into a host organism. And that's how scientists can give the host new characteristics or functions. That wraps up today's video. Thanks for watching, and if you would like the notes to keep studying, they're waiting for you in the description. See you in the next video.